In our last few lectures, we've looked at the 3D structure of DNA and RNA. Of course, we started with the famous double helical structure, which we use to hold all of our genetic code in our nucleus. But then we also looked at the variants of both DNA and RNA structure and how those may play a role in our transcription and translation processes. So, of course, as we saw with our proteins, these 3D structures really do have an incredible, important role in determining how these core biological processes take place. Now, one of the things that we saw with proteins is that 3D structure wasn't a permanent and immutable structure, right? So we could change our temperature or change our pH, and we could cause it to denature, and we could break that 3D structure. DNA can be denatured in very similar ways. So if we increase the temperature, or if we have large changes in pH, we can break apart our double helical structure of our DNA. So if we denature it, we can lead to a partially denatured DNA where we might have just some regions of our DNA where we pull apart those double strands and form just our free single-stranded DNA. Or we can fully denature it and create actual separate strands of DNA in just random coiled structures. Now, of course, remember that denaturation can be reversible. We refer to this as annealing in the case of our DNA here. So after we denature and separate either partially or fully our DNA strands, we can then anneal it and reform our double helical DNA. Our covalent bonds remain intact during this process. Again, remember, denaturation is just changing the 3D structure. It's not actually breaking apart our DNA here. And so that also means that our genetic code remains intact. So really what we're doing is we're just breaking those hydrogen bonds between our nitrogenous bases of our DNA and separating out those two strands. So we can actually then just reform those same hydrogen bonds after we lower the temperature again or return our pH to normal levels. And because we have that complementary nature of our DNA, we should be able to perfectly reform our original double helix of DNA. Now, our hydrogen bonds are not the only interaction, of course, that hold our bases together. Remember, we also have those hydrophobic bases coming together in the core of our helix, and the aromatic rings of our bases stack on top of each other to help increase the interactions there within our helical structure. And so this also results in a change in our UV absorbance. So the UV absorbance increases when our bases are free and don't have that stacking interaction, and it decreases when they are stacked closely together in our double helical structure. So if we're going to melt our DNA or denature it using heat, a thermal denaturation, so we would take our DNA, right, at standard temperature, it would exist as our double helix. As we heat it up, our two strands will disassociate, and then as we cool it back down, our two strands will re-anneal. We can reform those same hydrogen bonds and reform our double helix. So essentially, our DNA melts in a similar way to a liquid turning into a solid and vice versa. Right? We can take that double strand structure, melt it into our single strand, and then refreeze it essentially into our double helix. This reversible thermal DNA denaturation is actually very useful for us both in the body and as scientists. So in the body, we can use this sort of melting process to separate those DNA strands, so then we can use it as a template for copying our DNA during replication or transcription. From a scientist's perspective, this is also very useful in lab because, again, we can easily separate those strands and then copy them in order to create copies of our original DNA structure. So this actually allows for very useful techniques such as the polymerase chain reaction, or PCR, that's used to amplify different DNA sequences for various experiments. This thermal DNA denaturation is commonly monitored by UV spectrophotometry. So again, as I mentioned, your base stacking will be lost as you pull apart the helix and go to a random coil structure. and that change in base stacking will be reflected in a change in the absorbance. So we can actually determine essentially a melting temperature for our DNA helix by heating it and then just measuring our denaturation with that UV absorbance. We'll see that there will be a couple of different key factors that affect that melting temperature for our DNA. The first, of course, is the length of our DNA, right? So the longer our sequence, the more hydrogen bonds we'll have, and the harder that will be to break apart. Of course, we see this for all molecules in general, just because as it gets bigger, you're going to have more interactions between those molecules. So longer DNA would have a higher TM, represented here with the red graph, and shorter DNA would have a lower TM, represented here with the blue curve. Our melting temperature will also be highly dependent on pH and ionic strength. So remember that our DNA has a backbone formed from phosphates and sugars. Those phosphates, of course, are all negatively charged. And so we have negative charge repulsion between our backbone, which can act to destabilize our DNA. If we increase the salt concentration or increase the ionic strength, those salts can help to shield that negative charge and reduce the repulsion between those phosphates. So that will in turn strengthen our DNA structure, and it ultimately increases our melting point. pH, of course, you can imagine will affect our melting point as well, because it changes the charges both of our phosphates as well as potentially our DNA bases themselves. The third key factor that affects our melting point is actually the base composition of our DNA. So if we have a higher Cg content, we'll tend to have a higher melting temperature. So if you remember from our structures of our G and C, our guanine and cytosine, when guanine and cytosine bond together in DNA, they form 3 hydrogen bonds compared to the 2 hydrogen bonds of adenine and thymine, or of course, adenine and uracil. Because of this, our GC bonds are stronger than our AT, and we'll tend to have then a higher melting point for our DNA as we increase the percentage of G and C. Right? So you'll notice we have a linear relationship here where if we have relatively low GC content around here at 30%, we have relatively low melting point. If we have very high GC content, so up here around 70%, our melting temperature has increased by around 20 degrees. Obviously, of course, you'd have to be comparing these for similar lengths of DNA as well as at the same ionic strength, so that you don't have the other factors confusing your results here. Now remember, for our DNA molecules, our denaturation doesn't have to be fully denatured or fully folded, right? So we can actually have partially denatured structures, or we might denature in some areas of our long DNA strand, but not in others. And so if we take a large DNA sequence, what we'll see is that any AT-rich regions, adenine and thymine-rich regions, will melt at a lower temperature than our guanine-cytosine regions. So you can see it. This is a large DNA sequence here, as shown under an electron microscope. And you'll notice you have these little loops that form here as our AT-rich regions are denatured and separated, even while most of our sequence still stays in that double helical structure. So you see just the one double strand for most sequences, and then you see these occasional loops where the 2 strands separate when our AT content is higher. DNA denaturation is one way that we can change our 3D structure of DNA. In most cases, again, this denaturation isn't really a problem for us because we can simply reanneal our DNA to reform that original DNA double helix. The more concerning form of DNA structure changes occurs when we have chemical reactions or changes with our bases that would then lead to mutations, right? So because those bases would no longer pair up with their normal complementary partner, we then end up with incorrect base pairing, and we then introduce changes in our genetic code that will ultimately then cause errors in what proteins we make that we refer to as mutations. The first structural change that I want to look at that can cause mutations is tautomers. So tautomers are structural isomers that can interconvert between their two different structural isomer forms. Specifically, we're going to look at prototropic tautomers, which differ in the location of protons. So by an exchange of protons, we can convert from one to the other. Now, the most common form of prototropic tautomer is the conversion between a ketone form and an enol form. So this is something that we looked at extensively in organic chemistry because, of course, it affects the reactivity of our carbonyls. In the case of DNA or RNA, this ketone to enol tautomerization allows us to convert from lactam forms of nucleotides to lactim forms. So this will occur in uracil, as we show here. So we have, of course, two different carbonyls in our uracil. We also have our adjacent nitrogen and hydrogen here. And as we reform our tautomer here, so we would just have to transfer our hydrogen from the nitrogen over to the oxygen, and then shift our electrons to make a new double bond. So essentially our electrons are moving like this, as I've shown with my arrows here, and we convert to now the enol form shown here in the middle. So we have our keto form and our enol form. We could then repeat this as well on the lower half, right? So convert our ketone here to a second enol, which makes our double lactam form. So now both of our ketones are converted to our enols here. When we make these changes, you should notice that we've now switched from a hydrogen bond acceptor to a hydrogen bond donor for each of our carbonyls. We've also switched our nitrogen from a hydrogen bond donor now to a hydrogen bond acceptor. So whereas we initially had an acceptor, donor, acceptor pattern, now we have a donor, acceptor, donor pattern for our nucleotide. And of course, this completely changes the base pairing here, right? Normally, our uracil pairs with our adenine because our adenine has a donor and acceptor that pair up with the acceptor and donor of our normal uracil. So this tautomerization will lead to changes in the base pairing that forms between our nucleotides. So if we look at the single enol form of thymine, so thymine, of course, has the same basic structure of uracil, although note that our lower carbonyl doesn't normally form the enol form because in our nucleotide, our nitrogen is bonded to the sugar and doesn't have an extra hydrogen here. So we only switch our top two groups into the enol form, and so now we get a donor, acceptor, acceptor pattern, and this perfectly matches with our guanine. So guanine has an acceptor, donor, donor pattern, and we can base pair then between our enol form of thymine and our guanine. You can notice a very similar pairing over here in the lower right. So our guanine can also form a tautomer using the carbonyl on our guanine, and that will change its pattern to a donor, acceptor, donor. And this can match up with the original pattern of our thymine with its acceptor, donor, and acceptor. So again, if our guanine tautomerizes to its enol form, it will end up pairing with thymine instead of cytosine. Our adenine and cytosine can also undergo similar tautomerization, Although in this case, instead of a ketone rearranging to form an enol, it's our amine group rearranging to form this amino tautomer. So notice how we've rearranged the position of our hydrogen here on our adenine. So we moved one of the extra hydrogens from our nitrogen on the top down to our hydrogen here on the left side. And now we've created a acceptor donor pattern for our adenine that will match up with the donor acceptor pattern of our cytosine. We can also do the exact opposite and form our amino form of cytosine. We switch it now to an acceptor donor pattern to match up with the donor acceptor pattern of our normal adenine. So our base pairing is actually completely inverse if we have our amino or enol tautomer forms of our bases. So based on this, you might think that our tautomerization would completely ruin the complementary base pairing within our DNA. However, it's important to note that our enol and amino forms of our tautomers are very minor compared to our ketone forms, right? So we're much more likely to stay in that more stable ketone form, and it's only a very small percentage of our bases that would switch into this enol form. So we do see these tautomers creating pairing issues occasionally, but it's something that our DNA repair machinery can fix as it occurs at a relatively low rate. There are many other possible chemical reactions that can change our DNA structure as well. The next one that I want to look at is deamination. So deamination is a slow reaction that will replace our nitrogen groups on bases with a carbonyl. So in the case of cytosine, if we replace that nitrogen with our carbonyl, we produce uracil. Now, although this reaction is very slow, because we have such a large number of bases in our DNA, the overall effect is relatively significant. So there's approximately 100 cytosines converted to uracil every day within a given mammalian cell, right? That's just because we have billions of bases within our cells. And so even if just a couple are converting over to uracil, it ends up adding up over time. This deamination reaction actually shows us one of the advantages of using thymine in our DNA as compared to uracil. So if we use uracil in our DNA, then as we have this deamination reaction occurring, we would be producing more uracil, and we wouldn't be able to tell that uracil apart from our other uracils that are already in our DNA. So it would be hard to even notice that we had DNA damage here from our deamination reaction, because it would just look like normal uracil within our DNA. However, our thymine has an extra methyl group over here on the right-hand side. So if we convert from cytosine to uracil, we can immediately recognize that that uracil is not a normal part of our DNA, and we can then act to correct this mutation here caused by our deamination. So if instead we had methylcytosine being deaminated to thymine, there we have more of a concern, because now we can't tell that thymine apart from other natural thymine within our DNA. This deamination reaction can also occur for our other bases. So we can see adenine can deaminate this amine to produce a carbonyl, or guanine also has an amine that we can react, and we produce this hypoxanthine or xanthine from this deamination reaction. In both cases, our product base is not one of our 5 common bases, so there's no concern about our body not being able to recognize that new base and then, of course, correct that DNA damage there. In our deamination reaction, we are simply removing one nitrogen from our structure. A depurination involves hydrolyzing our N-glycosidic bond and completely removing the entire nitrogenous base. So as the name suggests, this is most significant for purines. So we can cut off our purine, either the guanosine or adenine, and you end up with your free base, and then your apurinic residue, where you just have the sugar with the free anomeric hydroxy group there that was previously connected in that N-glycosidic bond. So again, even though it's a relatively minor reaction overall, because of the large number of bases in our body, we tend to lose around 10,000 purines per day from this depurination. Again, our DNA has complex repair machinery, which can easily recognize that missing base. And so generally, this doesn't lead to a permanent mutation because our body can repair it. It's only if there's enough damage that it kind of overwhelms our repair mechanisms that you'll actually have those permanent mutations created in your genetic code. There are a variety of other reactions that can change our DNA as well, but these will often require addition of other agents in order to create the actual reaction. So if we have oxidative species, we can create a variety of different oxidative damage to our DNA. So we can hydroxylate our guanine, for example, and this will most commonly occur for our mitochondrial DNA. We can also have chemical alkylation, where we add on carbon groups. So for example, our guanine can be methylated. And again, our body would have to work to correct these modifications, and it would all be dependent on kind of if the chemical agent that's causing the damage is able to overwhelm your natural repair mechanisms. Here are some examples of some of the different alkylating agents. So in general, they're going to tend to have carbon groups that they can relatively easily donate to our base in order to create that alkylated nucleotide. Now, it's not only chemical agents that we have to be concerned about for causing mutations. So as we all know, of course, radiation can also induce mutagenesis. This can be in the form of UV light, which can induce dimerization of our nucleotides. This, of course, leads to skin cancers, or it can be in the form of much stronger radiation. So ionizing radiation, this would be X-rays or even higher energy gamma rays. And these would actually cause even greater damage. You can potentially break open your base ring or even break your DNA strand completely. The ionizing radiation tends to be the most difficult to fix because you're causing the largest change in your DNA structure. As I mentioned before, right, we can, of course, work to correct these modifications. It's just whether or not you're able to correct all those mutations. Over time, you'll tend to accumulate the mutations that you're unable to fix, and that then leads to some of the aging effects as well as, of course, cancer development. Here's an example of what our UV light-induced DNA damage looks like. So again, it's going to cause dimerization of pyrimidines. So here we have two adjacent thymines. And within our thymines here, we have, of course, our stacking between our bases. And we also have our double bonds, which are somewhat reactive. So our UV light can actually catalyze this reaction between our double bonds to produce this dimer here, where we've now linked our two double bonds into another cyclic system here. There are a couple different ways we can do this. We can also end up breaking that original cyclic system just to create a single linkage here. But the end result is that you have these two bases that are now linked together with a covalent bond. Now, of course, that bond doesn't immediately change our base pairing since we're not actually changing those hydrogen bond donors or acceptors. But what it does is it changes our overall 3D structure, right? So those two thymines are now kind of forced together, and that will affect our overall DNA 3D structure. So we end up producing this kink because our thymines can't quite rearrange themselves into the proper double helical 3D structure. So again, our repair machinery would be able to feel that difference in structure, since, of course, as we've talked about, that structure affects the interaction between proteins and our DNA. And so they would recognize that kink in order to try to repair and correct that mutation. So throughout this lecture, we've seen many different ways that we can change the structure of our DNA, both through temporary changes as well as more permanent mutations. Some of these can be very useful. So of course, thermal DNA denaturation allows us to pull those strands apart and then copy them, while others tend to be more just negative effects, causing mutations and potentially cancer. With all the different ways that our DNA structure can be damaged, it's actually quite remarkable how well our cell is able to correct that damage and keep our DNA functioning as our genetic code. So it's just another example of the incredible design in biochemistry that we have this machinery in place that's able to protect our genetic code and make mutations a relatively rare occurrence instead of something that happens hundreds of times a day.